Hello, this is a research proposal on a step towards plastic-free world by producing mass biodegradable fungi through replicating in hydrolase enzyme. The scholar name is Tafhim Islam. The scholar's affiliation is Youth Research Journal. Mentor's name is Sanaul Haq. Moving on to the introductory part. Moving on to the introductory part of this proposal. In the present world, one of the most harmful pollutants of the environment and a threat to our ecosystem is plastics. Some fungi are used by nature to mitigate environmental pollutants, and this process is called fungal bioremediation. Fungi, with the help of their mycelia and releasing hydrolase enzyme, degrade plastic. The purpose of our research is to replicate this enzyme in fungi which do not contain them so that we can limit plastic emission. We will move on to our research problem. The sole problem of our research is plastics. Plastics are one of the environment pollutants which are emitted from factories, hospital waste, farm waste, and many more, resulting in all elemental pollution and eventually climate change. These hamper our ecosystem by disrupting food chains and damaging soil fertility. Besides, they are one of the main reasons behind carcinogenic diseases, birth defects, neurological toxicity, genotoxicity, and many more because of the microplastics. All of this cannot be solved in an instant, but a leading step can be taken in this situation. Moving on to the research question. The main question of this research is, can hydrolase enzymes be replicated in fungi from species such as Pestaloteuthis microspora that do not naturally have them? Besides, can we enhance the plastic degradation via bioremediation, and can we mass reproduce this specific fungi? The research is divided into the following steps. 1. Identification of the perfect fungus for replication. Most appropriate to select in this case should be a healthy sample of Pestaloteuthis which was previously able to degrade plastic in a lab test. Second, extraction of hydrolase enzyme. The fungal enzyme can be extracted using ureal solutions in a controlled environment of a lab. Third, identification of the genome. In this case, specified genes are glycoside hydrolases, polysaccharide lyases, and carbohydrate esterases. Fourth, gene cloning. Using gene extraction, PCR amplification, and cloning vector, we will replicate the gene. Fifth, transformation into host cells. The extracted gene will be transformed into a suitable host, and we need to find a proper recombinant plasmid. Sixth, protein expression. We need to induce protein expression under optimized conditions and further follow cell lysis and purification techniques. Seventh, sustainability of the fungus with the new gene. We need to observe the fungus which will carry out the new gene for any other complications and see the adaptability in our environment. These are all the steps required for the process. Now we will talk about the significance of this research. The bioremediation fungi are one of the key aspects which are able to degrade fungi naturally without any significant chemical reaction, so it will be a stepping stone in redeeming our natural balance by eliminating plastic from our ecosystem. This research will provide a helping hand to introduce more of the plastic eating fungi. If successful, we can introduce these fungi in industries where they can control plastic wastes. The research will provide a better understanding of fungi genome as well. We can work on all sorts of plastic and they can be degraded. These can open up branches on medical and medicinal researches as well. We can be economically benefited from this research, and it will help to be an alternative for the costly methods used in our industries. The research duration will be within 1 year, where we will work on specific species of fungi and collect data on them so that we can analyze them. And there are lesser chances to fail since all the equipments and methodologies are known. The budget will be spent on limited aspects, so the budget will be brought to a minimum. I expect to publish this research paper in Youth Research Journal and practically use them in industries in a large scale. I will end the video with not any logical discussion or point of view. I'll just ask you to think of the turtles, dolphins, and birds who get stuck in polythenes or other plastic nets. I'll ask you to think about your child who will suffer from cancer or any other serious health hazard. You could have ended that. You could have stopped that. You could have taken a step which could have solved this issue and not only saved your children but also millions of plants, animals, and other living species, and eventually save our planet. I want you to give this research a chance to save our Earth, to let Mother Nature heal herself. Maybe it's not too late. Maybe it's not too late to heal our planet by stopping the emission of plastics. Thank you.